So here we have a new passage, and this passage is from biology. And as you see the past year CAT papers, you will find that plenty of passages come from natural science, and most of those passages are research oriented, in the sense that they have been picked from some kind of research journal or some kind of science journal, right? So we should be very good with these kind of content. And here is one passage that discusses a similar idea. It says here, after 30 years of investigation into cell genetics, researchers made startling discoveries in the 1960s and early 1970s, which culminated in the development of processes collectively known as recombinant deoxy. So we don't know how to pronounce this, but then it came to be known as rDNA technology for the active manipulation of cells' genetic code. So what happens in this first sentence, that during some period, 1960s and 1970s, so the researchers, they developed what we call as the process of rDNA, which is nothing but manipulation of cells' genetic code. The technology has created excitement and controversy because it involves altering DNA, which contains the building blocks of genetic code. Using rDNA technology, scientists can transfer a portion of the DNA from one organism to a single living cell of another. The scientist chemically snips the DNA chain of the host cell at a predetermined point and attaches another piece of DNA from a donor cell at the place, creating completely new organism. So we have read the first paragraph. Many of you must be familiar with this kind of topic. But what does this paragraph tell us? First of all, it introduces the idea of rDNA and tells us what this rDNA means. And he says that this rDNA is all about transferring one DNA from one living thing to another living thing and creating a new living thing. So that is what overall the idea is. That you have rDNA, so the scientist chemically snip the DNA chain of the host and attach it to another one and creating a completely new organism. So this is a very, very simple paragraph. Though too many words have been used to express the same idea, we know in short what exactly this paragraph has to say. Now let's go and check the second paragraph. Proponents of rDNA research and development claim that it will allow scientists to find cures for diseases and to better understand how genetic information controls an organism's development. They also see many other potentially practical benefits, especially in the pharmaceutical industry. Some corporations employing the new technology even claim that by the end of the century, all major diseases will be treated with drugs derived from microorganisms created through rDNA technology. Pharmaceutical products already developed but not yet marketed indicate that these predictions may be realized. Now, what do you see here in this paragraph? While reading, you must have realized that there are lots of positive things about rDNA. And obviously, they have to be positive because they are coming from proponents of rDNA. So here we have the word proponents, and proponents are always the ones who support something. It is the exact opposite of opponent, right? So the proponents tell us a lot of number of good things about rDNA. Let's not try to memorize how many good things and what those good things are. If a question comes on the proponents good things plan, they can always come to the this paragraph and look for the answer, right? So let's go and take the next paragraph. It says proponents also cite. So you have one more paragraph given to proponents. It says here proponents also cite non-medical applications for this technology. Energy production, it says here, energy production and waste disposal may benefit. Genetically altered organisms could convert sewage and other organic material into methane fuel. Agriculture might also take advantage of RDN technology to produce new varieties of crops that resist foul weather, pests, and the effects of poor soil. A major concern of the critiques of rDNA research is that genetically altered microorganisms might escape from the laboratory. Because these are microorganisms are laboratory creations that in all probability do not occur in nature, their interaction with the natural world cannot be predicted with certainty. It is possible that they could cause previously unknown, perhaps incurable diseases. The effect of genetically altered microorganisms on the world's microbiological predator relationships is another potentially serious problem, pointed out by the opponents of rDNA. Introducing a new species may disrupt or even destroy the existing ecosystem. The collapse of interdependent relationships among species extrapolated to its extreme could eventually result in the destruction of humanity. Now, what do you have here? The way we had the positives in the earlier paragraph, here we have plenty of negatives, right? So, and where does the negative start from? It starts from this point here, right? So, it says here a major concern of the critiques, right? So here we have the critiques of rDNA. And from this point till the end, we have the critiques of rDNA. And in the first part, we have the proponents. So in the earlier paragraph and here, you have the proponents and then you have the opponents, right? And then further, what do we have? Opponents of rDNA technology also cite ethical problems with it. For example, it gives scientists the power to instantly cross evolutionary and species boundaries that nature took millennia to establish. The implications of such power would become particularly profound if genetic engineers were to tinker with human genes, a practice that would bring us one step closer to Aldous Huxley's grim vision in Brave New World of totalitarian society that engineers human beings to fulfill specific roles. Now, we have finished the paragraph here. We know that the paragraph is primarily dominated by the word rDNA, correct? The author explains what rDNA is in the first paragraph, and after that, he goes on to give us plenty of positive things in the through the proponents. So the proponents have said plenty of positive things about rDNA. And then in the next paragraph, barring the first sentence, which talks about proponents further addition of something, then the criticism follows. So the critics have mentioned a few things about how this might be harmful and may not be good for the environment and for the future society. Now, the point is that when you read this kind of passage, you are likely to come across lots of details. I mean, if you say proponents, then the proponents have to they have their own opinion and they are giving two, three, four, five number of points. Same goes with the opponents; they also have to give 4 to 5 number of points. The idea is that you should not try to remember all these. Of course, when you see the options and when you see the question, it will automatically strike you as to where this has come from and whether this point had come in the passage or not. There are some students who try to read the passage only once and then mark all the questions in one go. That might not always be a good strategy. It can work if the questions are really very simple. But if the questions are not simple, then we'll always suggest that you read the question and check which part of the passage can have the answer to that question and then look at the options and compare. So the idea is that you should be sure about the accuracy of your answers. Sometimes just because we are in a hurry and we just mark the answers randomly, we we got many easy questions. We end up getting many easy questions incorrect, right? So if you check what is the detailing here, the detailing here is about the positive and the negatives of rDNA. Number of points have come, but then we haven't bothered about it. We know that okay, there are a few points in the positive, there are a few in the negative. If the question comes, we will go to that part of the passage and we will find the answer. So there is no need for you to worry about the intricate details. How we have mapped the key nouns? The key nouns definitely is the rDNA. And secondly, the positive things coming from rDNA and the negative things coming from rDNA. So there is a broad comparison going on. There is nothing like multiple things have come here and the author is discussing multiple relationships. So it is primarily about the pros and the cons of rDNA technology, right? And if you look at the gist of the paragraph and get the central idea, well, the central idea would be that rDNA seems to be a promising technology for many proponents, but the critics have their own concerns, and they have those are valid concerns, right? So this is the central idea of the passage. And then it says leave that part which you find difficult to understand. Now, not much has been difficult in this passage. Very much of it is very much clear, and it is meant for the layman like us. So this is what the whole passage is all about.